Hello everyone, welcome to Patho Excellence and Lymph Node Cytology video series. As we know, when we look at a lymph node FNAC smear, one of the biggest challenges is the wide differential diagnosis ranging from reactive conditions, infections to lymphomas and metastatic malignancies. So for many PG students, the difficulty is not in recognizing individual cells, but in answering a more important question: where do I start? So this is where the algorithmic pattern based approach becomes extremely useful, right? And instead of jumping directly to a diagnosis, this method teaches us to first identify the overall cytologic pattern at low power and then follow a structural diagnostic pathway. So by doing this, lymph node aspirates can be broadly classified into three algorithms. Each of these patterns immediately narrows the differential diagnosis and help us to choose the most appropriate ancillary investigations such as microbiology, IHC or flow cytometry. So in today's session, we will learn how to apply this pattern-based algorithm step by step, just like it is used in real hematopathology practice. So by the end of this lecture, you should be able to approach any lymph node aspirate in a systematic, confident and exam-oriented manner. So let us begin with the basic concept of pattern recognition in lymph node cytology. So whenever we assess our aspirated sample, we have to first assess adequacy. So before applying any algorithm, adequate sample must have sufficient lymphoid cells, minimal blood dilution, preserved cell morphology and representative lesion material. And suppose if inadequate aspirate is there, repeat aspiration is must. And now second step is, now you have to do primary pattern based classification. So at low power screening, lymph node FNA are must divided into three major patterns. So first, whether you find necrosis or neutrophils or granulomas. Second pattern is cell aggregate pattern. If extensive amount of aggregates are there, then you have to identify. And third is dispersed cells pattern. So this initial classification guide you to differential diagnosis and which ancillary test is required for further classification. Now let's see algorithm 1. In here, necrosis, neutrophil and granulomas we have to identify, and this pattern is called necrosis or neutrophil or granulomas pattern. So what are the cytological features? So you will find necrotic debris either in the form of caseation, numerous neutrophil if abscess is there, granulomas and epithelioid histiocytes and caseation, right? Now common diagnosis, when you find granulomas necrosis, it is tuberculous lymphadenitis. If neutrophils are predominant, suppurative bacterial lymphadenitis, some fungal infection, Kikuchi disease or necrotic malignancy. And in this pattern, most important ancillary tests are microbiological culture, AB stain, fungal stains or PCR for tuberculosis. So here in this smear, you can identify this is the necrosis and granuloma. This is the hyper view of granuloma where you can find these are the epithelioid cells. And if you do AB stain or Zern stain, you can identify this acid fast bacilli tuberculosis easily. So this is the granuloma pattern in algorithm 1. Now, conventional smear slide shows background where you can find necrosis, inflammatory debris, etc. But if you see liquid based cytopathology where you will have a clear background. So this is one type of drawback of this liquid based cytopathology, because you will not find any background details. Here cells are easily identifiable and here you can identify this mitosis, and here this is the crescentic histiocytes in smear. So, in algorithm pattern 1, when you find necrosis neutrophil granulomas, the ancillary techniques are culture, spatial stain, and if neutrophil and necrosis, suppurative lymphadenitis, and these are the differential diagnosis based on this feature. If neutrophils granuloma plus or minus necrosis, which is caseating type, then suppurative or granulomatous lymphadenitis, where tuberculosis or other fungal and cascade diseases in differential diagnosis. Granulomas and necrosis, it is necrotizing granulomatous lymphadenitis, where tuberculosis and non-tuberculosis as well as fungi are common along with syphilis. Now, granulomas without necrosis, then it is non-necrotizing granulomatous lymphadenitis. So, if benign disease considering, then sarcoidosis, any parasites, Crohn's disease or Whipple's or foreign bodies, silicone, etc., can lead to this granuloma without necrosis. And in case of malignancy or malignant lesion like squamous cell carcinoma, seminoma, thymoma, etc., which may be metastatic to lymph node and will lead to granuloma without necrosis. Hodgkin's lymphoma, B cell lymphoma, T cell lymphoma or tumor associated sarcoid reaction may lead to this granuloma without necrosis. So, this is important algorithm 1 pattern. Now, let us see algorithm 2, where you find cellular aggregate pattern predominantly. So, in cytology you will find cohesive cell clusters, architectural fragments, syncytial groups or tumor cell aggregates. So, common diagnoses include metastatic carcinoma, Hodgkin's lymphoma, anaplastic large cell lymphoma, where you find aggregates in abundance. So, if you find cell aggregate, try to make cell block and go for immunohistochemistry. If lymphohistiocytic aggregates, which is due to germinal center fragment aspiration, which is more common in reactive lymph node, and it is confirmed by numerous tingible body macrophages and polymorphous cell. If you find centrocytic centroblastic aggregate, think for follicular lymphoma and go for algorithm 3, which we will discuss in next slide. Histiocytic aggregates, which is common in granulomatous lymphadenitis or anthracotic lymph node, and if you find granulomas or histiocytic aggregate, follow algorithm 1. And epithelioid or spindle cell aggregate, then rule out metastatic sarcoma, metastatic carcinoma, melanoma or germ cell tumor, some benign epithelial or mesothelial and nevus cell inclusion and non-lymphoid target. And it is confirmed by and classified by cytokeratin, CD45 or S100 ISC markers. So, here this type of aggregates, if you can identify in low power, then go in search in high power view what type of aggregates is there. So, here you can identify these are the polygonal cells with cell enlargement, hyperchromasia and abundant cytoplasm, and this is squamous cell carcinoma in a lymph node having metastasis. Here the cell pattern is different, but aggregates are there. These are the cuboidal to columnar cells with palisading appearance, and some of the cells having nuclear pleomorphism and compact chromatin. So, this is a adenocarcinoma metastasis to lymph node. And sometimes in Hodgkin's lymphoma you will find this type of RS cells with lobulated nuclei and clumped chromatin and large nucleoli. So, this is a case of Hodgkin's lymphoma as well as if you find classic RS cells with binucleation and prominent nucleoli, this is again a classical clue to Hodgkin's lymphoma. Sometimes anaplastic large cells lymphoma show cell aggregate, but the cell is large or gigantic in nature, highly pleomorphic, cytoplasm is abundant bluish and vacuolation is easily visible, and nuclei chromatin is irregular and multiple large or macronucleoli are visible. So, this is a case of anaplastic large cell lymphoma which is ALK positive. Here also metastatic squamous cell carcinoma in lymph nodes. So, background shows lymphocytes, some polymorph, and these are the squamous cells which are malignant in nature, and background is showing inflammatory processes. And sometime you may find this type of tadpole cell where the malignant nucleus and cytoplasmic process, this is the pap smear, this is the Giemsa smear. So, in this type of algorithm pattern or cellular aggregate pattern, most important ancillary test is IHC on cell block. So, if you have a Hodgkin's lymphoma in differential diagnosis, go for CD30 and CD15. Anaplastic large cell lymphoma, CD30 and ALK, and metastatic carcinoma, you go for cytokeratin. Now, let us see algorithm 3, which is dispersed single cell pattern. This is most common pattern where you find predominantly single cells, lymphoid cell dispersion, minimal clustering, and here most important ancillary test is flow cytometry. So, purpose is to determine B cell clonality and immunophenotyping. So, when you find a dispersed cell pattern, so at the low magnification, you try to categorize whether the cells are polymorphous, where you find a tangible body macrophages and centroblast, centrocyte or immunoblast pattern, right? Or having small monotonous or monomorphic lymphocytic pattern, or another pattern is pleomorphic cell pattern which are large in size, irregular or not, or third is metastatic where highly cohesive clusters, 3D ball like clusters are there or not. So, from this pattern you can identify whether your cells having monotonous appearance or polymorphous appearance or pleomorphic appearance. So, this is the category or algorithm 3 where you have to categorize your dispersed cells into 5 categories, whether it is polymorphous and pleomorphic. So, what is the difference between polymorphous and pleomorphic? Where polymorphous means you will find all the lymphocytes in a different stages, like from starting from small lymphocytes to centrocytes, centroblast, immunoblast, tangible body macrophages, etc. Where in pleomorphic cells, all the cells having malignant look and they are having varying sizes, different sizes and shapes. So, when you find polymorphous, most common is reactive hypoplasia, infection related lesions, Castleman disease, Kimura disease, dermatopathic lymphadenopathy, Sézary syndrome, Katz-Reg, or some lymphoma like follicular lymphoma grade 2 or marginal zone lymphoma nodal type. Now, in pleomorphic cells, as we have seen already, like Ritz-Sternberg cells or anaplastic large cell lymphoma show pleomorphic large cells, or diffuse large base cell lymphoma or some metastatic carcinoma or sarcoma, right? So, polymorphous and pleomorphic cells where this type of appearance is there, where here you will find malignancy. Now, monotonous pattern is there. So monotonous again divided into small cell, medium size or large cell type. So monotonous most common in quotient benign lymph node, Castleman disease, small lymphocytic lymphoma, lymphoplasmacytic lymphoma, T cell or B cell lymphoma, Hodgkin's lymphocytic predominant type, right? Now medium size cell. So most common is DLBCL. So diffuse large B-cell lymphoma in medium size cells where follicular grade 1 as well as Burkitt's, precursor adult cell lymphoma, peripheral T cell lymphoma and Sézary syndrome. In monotonous large cell, so large cell you will find DLBCL common, sorry it is confused here, this is the here it is Burkitt and here it is a DLBCL. Here this monotonous large cell most common example is DLBCL, follicular grade 3, peripheral T cell lymphoma, granulocytic sarcoma, some nodal Langerhans cell histiocytosis, dendritic cell neoplasm or some metastatic carcinoma. And in this pattern, flow cytometry and FISH and molecular study is important to differentiate diagnosis. So, here again polymorphous, monotonous pattern divided into small, medium and large, and this is pleomorphic pattern. So, let us see all the pattern one by one. So, polymorphous pattern where you find small lymphocytes, centroblast, immunoblast, plasma cell or mixed cell pattern. Most common is reactive lymphoid hyperplasia. So, here you can identify these are the small lymphocytes. Here this is immunoblast, largest cell, centroblast and cleaved nuclei are seen in the centrocytes. So, polymorphous these are the causes where reactive and infectious etiology or syndromic or neoplastic etiology categorized in this. So, diagnostic polymorphous presentation are uniquely dominated by reactive hyperplasia and infectious compared to other morphological categories. Monotonous where you find uniform small lymphocytes. Most common lymphomas are small lymphocytic or CLL, mantle cell or marginal zone lymphoma. So, here monotonous small cell pattern where you have to compare with the RBC. So, you can easily identify this is the small cell pattern. So, here these are the monotonous small cell pattern in the MGG stain. So, monotonous small cell pattern mostly SLL is the commonest, but these are the other condition you can find monotonous small cell pattern. Now, medium cell pattern. So, medium cell pattern which is medium sized cells with basophilic cytoplasm and typical disease in medium size is Burkitt lymphoma. So, here you can identify this is RBC and you compare the nucleus of this medium cell. So, this is 1.5 to 2 times large and here in Burkitt's lymphoma there is cleaved nuclei. So, here you can identify these are the medium sized cells and having blue cytoplasm with vacuolization. So, this is the Burkitt lymphoma. So, these are the differential diagnoses when you find monotonous medium sized cells where follicular lymphoma grade 1, Sézary syndrome along with this all different types of lymphoma, but Burkitt lymphoma is common. So, here this is the metastatic entry point and this category marks the first morphological appearance of metastatic small cell malignancies which requiring non-lymphoid differentials. So, sometimes this type of cells may be present in this category. Now monotonous large cell pattern. So this is DLBCL type common example where large atypical lymphoid cells. So you can here easily identify RBC size and small lymphocytes, and you can compare. This is the 3 times larger cells compared to this mature lymphocytes. So this is the monotonous large cell pattern. So here these are the large cells. See, these are the background lymphocytes, RBCs, and size is comparable. This is a 3 times large cell. So diffuse large cell B lymphoma here shows highly cellular smear with large atypical cells, nuclear pleomorphism, and high NC ratio. So in this category, primarily if lymphoid neoplasm, if we see DLBCL, grade 3 follicular lymphoma, transform SLL, blastoid pattern of MCL, peripheral T cell lymphoma or natural killer, or some granulocytic sarcoma and dendritic cell neoplasm, and metastatic mimic where metastatic carcinoma, melanoma, sarcoma and seminoma need to be ruled out. Now, last important pleomorphic or anaplastic pattern where you find highly atypical large pleomorphic cells, and possible diagnoses include anaplastic large cell lymphoma, metastatic carcinoma, high grade lymphoma. See, this is the normal lymphocytes RBC, but these are the highly atypical large pleomorphic cells with cleaved nucleus, multiple macronucleoli. So this is the case of anaplastic large cell lymphoma. This is the mature lymphocytes. You can compare this cell with this large anaplastic blastoid cell of ALCL. So this is anaplastic large cell lymphoma smear. So highly atypical and pleomorphic enlarged cells. So, it is defined by wildly varying atypical and bizarre cellular morphology, and Hodgkin's lymphoma, anaplastic, diffuse large B cell lymphoma variant show this type of cells, and in metastatic pattern mimics are metastatic carcinoma, melanoma, sarcoma, seminoma. So, this is the shapeshifter diagnostic table where you can find in Castleman, Caesarean and nodal MZL where polymorphous pattern is there, and in Hodgkin's classic and DLBCL variant show pleomorphic pattern, right? And in uh, small, medium and large cell you can identify in small cell pattern lymphocytic predominant Hodgkin's, Castleman disease, or histiocytic rich or T cell rich diffuse large B cell lymphoma show small cell pattern. Medium size pattern is mostly seen in nodal marginal zone, Caesary and MCL, where large where you find transformed SLL, blastoid MCL or DLBCL shows this type of pattern. So, this is the differential at a glance where dispersed cell, whenever you find, go for flow cytometry or fish molecular study so that you can easily identify whether it is polymorphous, monotonous or pleomorphic pattern. So this is a unified structural model for hematolymphoid categorization. You copy this slide to categorize or to differential diagnosis your smear whenever you do microscopy with the this algorithm pattern based approach. So to conclude, the algorithm pattern based approach transforms lymph node FNAC interpretation from a confusing exercise into a structured and logical process. So always remember the sequence. Assess adequacy of the sample first. Second step, identify overall psychological pattern at a low power view. And third is classify the aspirate into whether it is necrotic or granulomatous or cellular aggregate pattern or dispersed cell pattern. And use this pattern to guide your differential diagnosis. Select the appropriate ancillary test And this approach not only improves diagnostic accuracy but also helps avoid common pitfalls and misinterpretations. So most importantly, it reflects how experienced cytopathologists actually think at the microscope. So next time you examine a lymph node FNAC, don't start with the diagnosis, start with the pattern. So thank you for watching. If you found this session helpful, please like, share and subscribe and stay connected to more high yield pathology content for PG training. So in next topic we will discuss the polymorphous lymphoid cell pattern in detail, including all the differential diagnoses. So keep watching and have a good day.